Dzień dobry, witam wszystkich na trzeciej edycji webinaru Wiedza ma warstwy, webinarów o nowych technologiach. W tej edycji czeka nas sporo ciekawych rozmów z liderami z rynku nowych technologii. Także gorąco zachęcam do odwiedzenia naszej strony sygnis.pl/warstwy. Tam możecie zobaczyć, co jeszcze na Was czeka w tym tygodniu. Na każdy webinar obowiązują zapisy przez formularz też podany na stronie. Dzisiejsze spotkanie będzie prowadzone w języku angielskim. Po prezentacji zrobimy sesję Q&A. Wszystkich gorąco zachęcam do zadawania pytań na czacie albo zgłoszenia chęci wypowiedzenia się na czacie, wtedy można będzie zadać pytanie live. Now we'll continue in English. So welcome once again everyone in our webinar series Knowledge Has Layers. As I mentioned in Polish, feel free to write your questions on the chat. In the end of the presentation, our speaker will answer them all. Or if you want to ask it live, please write it also in the chat, and then I will give you the mic and you will ask the question. So during presentation also, please mute your microphones. And now we will go to the most important part. It's a pleasure to introduce our today's speaker, Camille. I won't talk so much, just giving the microphone to you. The floor is yours. Sorry, but you're muted.
Sorry, Anna, I thought you were able to unmute me.
No, right. I can only ask you to unmute. Okay, now the floor is yours. Yeah.
Okay, perfect. Sorry about that. Thank you so much for the introduction. So the presentation is not going to be as long as an hour. Don't worry about it. It's not going to be super boring. I mainly wanted to talk to you about a few things. So mainly about the accurate isolation and quantitation or characterization of nanobiological particles, which I think is your main topic today. So what are the challenges in the field, and most importantly in the field that you guys are interested in? So there are like two main challenges, being the lack of standardization, whether we talk about extracellular vesicles or viruses or bacteria or just nanobiological particles in general. We lack of standardization in isolation and characterization. So why is that? Just because the measurements for those particles are typically done using optical techniques, such as DLS or NTA, so nanoparticle tracking analysis, that I'm sure you're all aware of, which actually is a major part of the problem. So you really need to be super careful with the kind of techniques that you're choosing to conduct your research. Otherwise, you can miss quite a lot of information. A good size measurement is really, really important, as I'm sure you're well aware of. And it's actually very easy to do, but again, that depends on the kind of technique you're using. One very important point that I want to bring your attention on is that resolving nanobiological particles in general, whether we're talking about small viruses or exosomes, extracellular vesicle, you need to resolve these small particles from the background noise. Otherwise, you're losing information on small particles. You need to resolve them correctly to differentiate them in terms of shape. So, for instance, you know that an exosome is a spherical shape, and some sort of viruses would be like more rod shape. So it's important to distinguish particles depending on their shape, and that is only done easily, if I may say, if you have a good pure sample. And that's another aspect that we will be touching on a little bit later in the presentation. I will first focus on the characterization side of things, and then I will quickly move on to the isolation side of things. So what are the solutions that we, Eison, as a company, would actually provide you with? So just extremely quickly, that's not super important. I'm sure you can find information if you want to browse our website. But Eison is a company that comes from New Zealand. So we've been around for 16 years now, and we're like the company of reference in the EV field for both isolation and characterization of viruses and extracellular vesicles. So the European headquarters are located in France, in Lyon, down south, which is where I come from. And we work with Sygnis and the team as they would distribute our products in Poland. But I'm sure you already know that. So as part of a solution, we have came up with different platforms as a product. The first platform would be the isolation platform. So as I told you, in order to get a good characterization of particles, you need to have a very good isolation method that's in place. So we're talking about viruses, VLP, so virus-like particles, viral vectors, extracellular vesicles, and you need to have those as pure as possible because it's going to make your life a lot easier later on, whether we're talking about vaccine development or gene therapy or diagnostics. So with our platform, Eison's platform, QEV isolation columns, and combined with our characterization technique, TRPS, which stands for Tunable Resistive Pulse Sensing, those two platforms combined together, they would provide the only standardized method of isolating and quantifying virions, VLPs, and extracellular vesicles. So the advantages of using that platform simultaneously all together would be that it's actually super fast, it's very simple, and it's very reproducible because we have moved on to the automated side of things. So as the characterization of samples, you actually only need 35 microliters of sample to be able to read your samples, I must say. And because you're having a real-time analysis that enables you to assess correctly some sample aggregation over time, if that's necessary. So just a very quick overview of what TRPS is. So on the right corner, you would see a screenshot of what's happening during a quad model sample characterization. So those would be just 4 different particle sizes mixed together and analyzed simultaneously. So TRPS, as I told you before, that stands for Tunable Resistive Pulse Sensing. It's a technique that enables the users to characterize particles ranging from 30 nanometers up to 15 microns. So you have quite a decent size range to play around with. So I have focused today's presentation with examples around the nanobiological field, so around extracellular vesicles and viruses, but in reality, whatever falls within this size range, 30 nano up to 15 microns, and whatever is able to be dispersed in electrolyte, I will touch base on that in a minute, you can measure. So we're working on a daily basis with liposomes, for instance, with paint ink or with other metallic or magnetic nanoparticles. Doesn't really matter as long as it falls within the size range, and 2, it can be dispersed in an electrolyte. So you might have heard about our competitors, let's say, so the optical techniques that I mentioned previously, so DLS and NTA. Actually, TRPS, that's the technique that fast surpasses those other optical techniques by 2 reasons. So one of the reasons is by being able to complete simultaneous measurements. So simultaneous measurement being the characterization of size and size distribution, concentration, and zeta potential of particles, because we use a particle by particle approach. And while the particles are getting through a consumable that we call a nanopore, so it's this is a little funnel through which the samples are going, and once they're going through that funnel one by one, they're being counted. So this is how you're getting a concentration. So we're getting independent data on each and every particle, and that enables us to get a multiplex analysis like the one that you do see on the screen. So I will explain a little bit further, but just really quickly, the red dots that you do see on your screen, so each dot is one particle, and each yellow peak here, that's a particle as well. And the bigger the peak, the bigger the particle, the smaller the peak, the smaller the particle. So that would be one population of big particle. That would be a second population of smaller-ish particle, and a third population and a fourth population of very, very small particles. So yeah, each red dot would be one particle. Each yellow peak would be a particle as well. It means the same thing. It's just another representation. So about the concept, how that works. So resistive pulse sensing is an impedance-based measurement. So TRPS technology, well, it's not very new. We've just rejigged it to modern times and being able to analyze smaller particles. But in reality, it's a technique that derives from the Coulter counter. Maybe you're familiar with it. A Coulter counter is an instrument that used to be used for cell analysis. So we've just taken that technique and rejigged it, and we were able to measure even smaller particles. So we're measuring the impedance of particles or the resistance of the particles. So how that works, we're working with this consumable that is called a nanopore. So it's a plastic membrane that is flexible because it's made of plastic with a hole in the middle. There is just one hole in the middle, and each and every nanopore is optimized to be able to measure a certain size range of particles. For instance, the one that's on the screen is called NP200, as you can see here. So that's the size of the nanopore is called NP200. And that would allow for the measurements of particles roughly between 80 nanometers up to 500 or 600 nanometers. So that means that you still need to know a little bit about your samples in order to select the correct nanopore. Because if you're taking a nanopore that is too big for your particles, the particles are going to fly through the nanopore, but you're not going to be able to see anything. The peak, so do you remember the yellow peaks that I showed you just in my previous slide? The yellow peaks would be lost in the signal to noise, and you're not going to be able to see them if your nanopore is too big. The signal to noise ratio would be too big, so the particles would be lost. On the contrary, if you're choosing a nanopore that is too small for your particles, you have very good chances to actually block your nanopore, because the particles are going to try to cross the nanopore, but the aperture is going to be too small, so they're not going to go through. So you still need to know a little bit about your sample to select the correct one. So as of now, with that, we have an entire range of 11 or 12 different nanopores, each of them being optimized for a certain size range in order to cover the entire 30 nanometers up to 15 microns. Of course, if you're working in a field and that requires analysis of both 30 nanometers up to 15 microns, you're not going to need the 11 nanopores, of course not, because they all overlap with each other. So some of them are really narrow, some other are actually really wide, and that all depends on the applications. And this is how we're here for, just to help users selecting the correct nanopores. In reality, to cover this entire size range, like 3 nanopores would be enough. But that's another topic. So as I told you, those nanopores are consumables. That means that they're not going to last forever, but you don't have to change them every day either. Obviously, it depends on the intensity of your work, the intensity of your measurement. We like to say that each nanopore would last for around 10 measurements. So it's true that if you're measuring 10 different samples on a single day, then yes, you're losing 1 nanopore a day. Some of them, like the bigger nanopores, would be a little bit more robust than the smaller nanopores. So depending on how clean your sample is and depending on how good you're taking care of your nanopores, you can actually prolong the lifetime quite a lot. So that's entirely up to the users to do that. But as a minimum, you can do up to 10 10 samples per nanopore. And then the software kind of helps you know if the nanopore has been overused already, if it's too dirty, or if you can still use it. So that being said, I want to explain very quickly the principle. You'll see that it's rather easy. It might be completely different to what you've been used to, but so you're having that nanopore that is stuck in between two electrodes and immersed with electrolyte from both sides. So when I'm talking about electrolytes, I don't mean any crazy electrolytes out there, like PBS is fine, NaCl is fine, KCl is fine, HEPAS as well. Doesn't really matter as long as it's conductive and it contains some salts. So we're usually working on a physiological strength buffer to mimic as much as possible what's going on in the human body. So you're having that nanopore stuck in between two electrodes immersed with electrolyte, and you're creating an ionic current through the nanopore. And because all of your particles are either positively or, most importantly, negatively charged, they would travel through the nanopore one by one. And as they travel through that nanopore, they're disturbing temporarily the current, and they're doing like a drop in current, which is a representation of the yellow peak. So again, remember, each peak is one particle. The bigger the peak, the bigger the particle. So this is what's happening. You're having a particle crossing the nanopore. It generates a drop in current, and it's being counted as a particle. Easy peasy. So as of now, so this is what you're seeing on the screen now is the instrument that's been the instrument of reference until very recently, which is called the Q-Nano Gold. So it used to be the gold standard for high precision measurements. It's a very good instrument. It gets reproducible and rather easy measurements, but we've heard from a lot of our users that the usability can be challenging. Why? Because a high sample throughput requires a certain level of expertise, and a lot of users would require a steep learning curve to become a good user. So some of you might know about that Q-Nano, have maybe used it already, or you might have heard that, yes, it's a good instrument, but it's not that easy to use. And it's true. You need to be, so first of all, it's like exactly like when you're learning on how to drive a car. You need to have your driving license in order to drive a car. That's a little bit the same. You cannot just say, oh, I'm going to be using that instrument without being trained on and without being certified on the unit. So that's the very first step. You need to know and understand what you're doing. And that goes through a proper training that is made by us, by the company. So just really quickly, what's happening when you're having that particle crossing the nanopore? So you do see a peak on your screen. So the length of the peak, as I told you, represents the particle size. When you're zooming in, the width of the peak at its base is the electrophoretic mobility, which is used to calculate the charge of the particles, or the zeta potential analysis. So that means the same thing. Charge and zeta potential equals the same. And the number of particles is the number of particles per screen or per minute, which is a rate, is your concentration. So, we know that it's been a little bit of a challenge to be using that instrument. So in order to address those issues, we came up with this new little instrument that's called the Exoit. So this is just quickly a picture of the Exoit, which is the big brother of the Q-Nano, and which, as we like to call it, represent a new era in single particles analysis. So what you do see on the computer screen here is just the user interface. So that instrument, the Exoit, is focused on usability, because remember, I told you the Q-Nano was great, but we've heard our users when they said it's a little bit too complicated to use and to understand. So the Exoit, to address that, is used on two things. First of all, a significantly enhanced ease of use for both new and experienced users, and two, a significant efficiency gains in terms of time and effort that's required to make measurements. So that means that you have real gains in precision and resolution, because if it's easier to use, then you're having a better throughput for results. And that also means that more users in the lab are going to be able to use the instrument. Maybe you know in your lab, maybe you have some instruments that are not so easy to use either, and there's just a fraction of your team that is able to use them, which is a shame if that specific person leaves or is on holiday or just leaves the lab to go to another position. Then the knowledge goes with them, and that's not okay. That's too bad, because it means that you need to reinvest a lot of things, not to mention just time and money, but you just need to reinvest quite a lot of efforts. And that's not happening anymore with the EXO-8, because both the instrument and the user interface, the software, are super user-friendly. So the knowledge doesn't leave with the user, because the instrument and the software guides the user's hand, it takes the user's hand through every single step of the way. So along with this comes a real gain of time, of precision, and resolution, because you're moving away from the potential human error that you used to have using the Q-Nano, which was the manual version of the EXO-8. So EXO-8 and Q-Nano, they would both be the same kind of instruments because they're using the same technology. They both deriving from the TRPS technology, but one is the manual version, which is the Q-Nano. The other one is the semi-automated version. So that is just a screenshot again of what's happening when you're starting your measurement, but I have a few videos that explains how exactly that works. So this is just a result of a tri-model sample analyzed using the EXO-8. So as you can see, you have the concentration versus size that is displayed as a histogram. So that's a tri-model sample made on purpose, where you have one population around 70 nanometers, another population around the 100 nanometer mark, and then a bigger but less concentrated population around the 150, 160 nanometer mark. So that has been done with only one nanopore, of course. So if you remember, each nanopore, they would be quite vast in terms of the size that they can actually measure. So you can actually measure some heterogeneous samples over a quite wide size distribution without the need for multiple measurements or without having to adjust the settings in between. That's specifically important when you're working with polydispersed samples, because working on polydispersed samples can be quite of a challenge, where you have subpopulations, either the smaller populations or larger populations, that can be identified with a high resolution and quantified individually. So it's great to monitor or to follow particles aggregation over time, for instance. The way it works is that it's a calibrated method. So that means that you're always comparing your unknown sample with the known calibration particles of known size, concentration, and zeta potential. So the software just really overlaps your sample run that you don't know with the calibration run that it knows. You do have to tell the software, that's my sample, that's my calibration, but then it does the trick. So that's a video that I mentioned just to show you how it works. So it doesn't have any sound on, unfortunately. I was hoping it would, but it doesn't. So I'm just gonna talk over it. So actually it does have sound. So, all right, so the measurement has started. As you can see, so this is the signal trace, and each peak here is one particle. So that is the software interface of the EXO-8. Each peak is one particle, each red dot is one particle as well, and there you have so the current expressed in nanoamp, so nanoampere, doesn't really matter, but this is what I was explaining when I was talking about the electrolyte that was used. So the higher the conductivity, the higher the current, the lower the conductivity, the lower the current. And you can monitor in real time what would be the shape of your histogram. So in here on the right of your screen, you do see one very, very narrow histogram, that means that you're working with a monodispersed sample. And as you can see, pretty much all of the peaks here have a similar size. So that was a calibration run, so it's a good thing that they have a similar size. I'm just going to go back a few seconds. So yeah, your calibration needs to be similarly sized. So see, the software here guides the users. Oops, there you go. The software here guides the users so that the user knows where in the measurement it is. So in this specific example, let me just pause it. In this specific example, we were running, so as I told you, a calibration sample. I think it was a 100 nanometers calibration sample. And we asked the software to perform like the 3 different measurements. So at 3 different pressures. So the pressure is one of the parameters that is automatically adjusted by both the software and the instrument. So it tells you where in the measurement you are at. So on the last run of the 3 runs total, now it tells you that, well, you're almost there, because see, it's very, very fast. The speed of a measurement would depend on how concentrated your sample is to start with. Now see, the software tells you calibration complete. Do you want to proceed further? If so, yes, replace the sample and flush the nanopore, make sure that it's clean. So that's very important as well, because it means that the software includes some sort of a quality control procedure to make sure that you have the most clean nanopore possible between your calibration sample and between your real sample, and between sample and sample. So that's essential. This is another video. Go. So that is another video that says pretty much the same thing. So see, we did already pressure 1. We have just done pressure 2, and we're going to be doing pressure 3. So now you can see that the XO8 auto adjust to another pressure. You don't have to do anything, but it's just that multi-pressure calibration step is very important when you want to do a good concentration measurement. So again, it tells you like you're 50%, close to 50% from the very last measurement. If you need to do some troubleshooting of any kind, you can just pause the analysis, do your troubleshooting, and resume the analysis, and it's like nothing happens. And as you can see here again, calibration is now complete, and the software asks you to move on to the next step. So I think you got my point. So moving on to the actual sample, because previously that was the calibration. So similarly, like the calibration, you're actually having a bimodal sample. So you do see that thanks to the size of the peaks, so the bigger peaks are bigger particles, smaller peaks are smaller particles. So if you take a look at there, you would actually see that you would have different sizes matching the different peak sizes. So I'm just gonna leave it for you to contemplate. So it definitely provides a reliable method to accurately calibrate your particles and your samples without any worry, because it does everything on its own. It just tells you, now put the calibration, now clean it, now put the sample. So see, as I told you, it was a bimodal sample. You can actually clearly see that it's a bimodal sample. That's one population here and the second population here. Obviously, in terms of concentration, you do have more of the bigger particles than the smaller particles, so it's normal then concentration-wise, the bigger particles are in dominance according to the smaller ones. And similarly, it would tell you like you have 100 particles more to count before I can reliably stop and provide you with a good sample measurement. So moving on to a quick comparison of the different sizing techniques that are available on the market. So there have been multiple publications over the past few years that were comparing TRPS with DLS, NTA, and DCS. If you're interested, I can give you those publications if you want to look at it. But in essence, those are just the results of comparison studies that have been submitted either a few years ago here on the left and on the right, just back in January a few months ago. So in which the most used techniques within the nanofield were used to analyze a quad model suspension. Here that was a tri-model suspension. Here that was a quad model suspension. So it's pretty obvious then the optical techniques, as you can see here, DLS and NTA, they cannot resolve complex samples, neither size distribution or concentration, because you're getting blinded by the bigger particles to the smaller ones and you cannot see the full picture. So that's why it's really dangerous when you're working in your field to stick to one optical technique, because it doesn't provide you with the full picture. Whereas TRPS, as it's here, being a real particle by particle number-based distribution, which does provide realistic and accurate measure of particle size, distribution, and concentration. If you're just strictly looking at this graph here, you do see that, so that was a quad model sample with similar concentrations that the NTA weren't able to distinguish because of a lack of resolution, which is a little bit embarrassing when you're working with polydispersed samples all day long. So that's why I'm insisting in be careful with the kind of techniques that you're actually choosing for your research. Moving on to the isolation side of things. So in order to get a good quantitation, a good sample analysis that's done, you, of course, need to have a reproducible and standardized isolation and purification method in place as well. So we have teamed up almost 10 years ago, I want to say, 8 to 10 years ago, we have teamed up with ANC, so the University of Amsterdam, to come up with what we call the QEV columns. So I'm sure you know about them. You would have heard about it already. So the size exclusion chromatography columns are just based on gravity flow. It provides a very fast separation time, so it takes about 15 to 20 minutes maximum, depending on what's the volume that you need to purify. It removes over 99% of contaminating background proteins. It would remove around 95% of contaminating HDL. As of now, our gel is Cephros gel. I'm not going to go into too much details because I'm not able to tell the secret recipe behind it, but it's not just Cephros, obviously. And we will be moving away from Cephros later in May to be able to work in another matrix inside of the QED columns and to get rid of the LDL as well, which is not possible as of now because we're only based on size difference, but LDL would be able to be captured by the gel and then released a little bit later. So anyway, that technique is really gentle because it would retain the structural integrity and biological functions of EVs. We have a range of 5 different column sizes, depending on what's the size that you need to purify. So we can purify as small as 100 microliters up to over 100 milliliters. And we're working now with a column that would be able to purify up to 1 liter. So again, it really depends on what your application is. We have 2 size range exclusion available, either 35 or 70 nanometers to exclude particles below 110 nanometers and other below 330 nanometers. All of them, regardless of the size, would be manufactured according to the ISO norm 13485. Forgot to mention just before that the 2 systems that I showed you, so the QNano and the Exoit, they are GMP. So then you get the IQOQ version of things available as well if you need to. What you do see here is another instrument, which is another semi-automated instrument that is called the AFC, for automatic fraction collector, that is only to be used with our QED columns that I just showed you. So because it's automatic, a little bit like the exotic, they look alike, both of them look alike. Because it's automatic, it allows you for the standardization, a customization if you're not happy with whatever by default parameters are set. It would provide you with some repeatability and convenience, of course, because the user just sets the parameters, go away and come back, and the isolation is completely done instead of doing things manually. So on this slide, I know it's very busy, it's a very busy slide, but here I just wanted to show you an example of how a standardized experiment using the Eisen products look like. So Eisen products meaning the AFC that you just saw, the QED columns that it was working with, and the exoid as a result for the characterization. So first you need to choose your biological fluid. Could be cell culture, plasma, urine, tears, whatever, and then you can move on for the purification part to the AFC to collect precisely each fractions containing the extracellular vesicles or the viruses during a QED isolation process. And then you can use those very pure fractions to perform a TRPS analysis to know the size distribution, concentration, and charge of those particles. And then, if you fancy, you can actually perform further analysis as proteomics or lipidomics and go even deeper and use our QED RNA extraction kit that we've just released as well, or perform a qPCR, for instance. So, as I just told you, and I'm going towards the very end of my presentation, we knew and we listened to the world when we heard that it was a wide need of reproducibility that was required from the research world, and that was requiring some standardization of methods. So it all starts with sample preparation and purification, and most methods will provide significant variation in results, even if the measurements that you do seem accurate. So we have chosen to position ourselves through the size exclusion chromatography columns that offer a very simple, efficient, and standardizable solution for purification. And further down the line, TRPS always being calibrated. That's like super important. There's one thing you need to remember is that calibration is key. You cannot guess without a calibration. It needs to be done a certain way, and you need to use calibration. Otherwise, you cannot get an accurate result. So because it's calibrated, it always offers a very simple and effective method for standardization. And of course, if you want to go a little bit further, the RNA extraction kits to get rid of all that would be of great help. I think that's it. Thank you so much for your attention. And if you have any questions, please feel free to ask me now.
Thank you so much for amazing presentation. And yeah, feel free to ask questions on the chat, or you can ask it live. So there is the right moment for this. Also, maybe if you will have some specific question later, I guess you can write to Camille. Here is her email.
Absolutely. Yeah, definitely. If you don't have any inspiration now, just feel free to email me a little bit later. That's my email address. So I think that's one question in the chat.
No.
That's you.
That was just me. Yeah, information about the start of the webinar. Okay, I think we will finish. So yeah, please remember that you can write to Camille or to me with your questions. I will direct it to Camille, or you can write directly to her. Thank you so much for sharing your knowledge with us for this presentation. And I want to welcome everyone to the next webinars that will take place on Wednesday. feel free to visit our website and see what upcoming topics are waiting for you. Also, please visit the ISON website to see what they have. Amazing information. Thank you so much. And yeah, everyone, have a nice day. And see you on the next webinars.
Thank you, Anna. Bye-bye. Have a nice day, everyone. Cheers.